So welcome everybody. My name is Kelly Henwood. I'm with the WSU Extension Regional Small Farms Program. And our program is based out of Clallam, Jefferson, and Kitsap counties on the Olympic Peninsula. And tonight I will be sharing a little bit about our Zoom format tonight, and then I'll turn it over to our guests to introduce themselves. So please utilize the chat function. If you have questions, save it till the very end. I'm sure there will be time for questions and answers and some discussion. You're welcome to either use the chat for your question and we'll monitor it, or keep it and ask it at the end. If you are not able to stay for the entire webinar, please just let us know. We have an evaluation link that we'll post in our chat towards the end that we'd love your feedback to demonstrate the importance of these educational opportunities. And if you have any other questions about our Regional Small Farms program or want to learn more about on our upcoming offerings that we have and you're not subscribed to our newsletter, we can add your email to our agriculture newsletter. We post a lot of events and host a lot of these opportunities for farmers in our region. So I'm going to pass it over to my colleague Laurel Moulton to introduce herself and then introduce our guest.
Hi, everybody. Welcome. I'm so glad you're here tonight. Um, I'm Laurel Moulton. I'm a horticulture and IPM specialist with the WSU Regional Small Farms Program. I'm based in Port Angeles, but serve Jefferson and Kitsap County as well. Just as a reminder, with our Regional Small Farms Program, we have all sorts of resources online, but we also have technical assistance available. You can go to our website and request technical assistance using the portal there, and we'll help you out. Um, Aside from that, it's my pleasure tonight to introduce our speaker. Our speaker tonight is Lisa Wasko DeVetter. She'll be talking about future-proofing small fruits, climate change, and mitigation. Lisa is based at the Northwestern Washington Research and Extension Center in Mount Vernon, where she leads WSU's statewide small fruit horticulture program, focusing on the big four fruits: blueberries, raspberries, blackberries, and strawberries. We're really excited to have her here today, and she occasionally comes out to visit farms in our area, so that's even better. Um, in the 12 years that Lisa has been with WSU, she's developed a diverse research and extension program with an emphasis on maximizing productivity, fruit quality, and on-farm efficiencies while ensuring the health of adjacent natural resources that are critical for small fruit crops. She's currently working on optimizing pollination services for small fruits, nutrient management, improving the end-of-life management of agricultural plastics used in small fruit crop production, improving machine harvesting technologies, and finally understanding and mitigating the stresses caused by climate change. And that's what she'll be talking about today. So with that, I'd love to turn it over to Lisa, and welcome. We're glad to have you here.
Great. Well, thank you, Kelly and Laurel. It's my pleasure to be here. And I don't do all of that by myself. I just want to acknowledge that I have an incredible team of staff, including graduate students, postdocs, and technicians. I'm really grateful for them. And, I'm going to be highlighting some of their work here. So let me just go ahead and share my slides and make sure they're coming through nicely. Laurel or Kelly, could you tell me, are the slides coming through nicely, or are you seeing the slide notes?
I'm seeing the slide notes.
I told you. It likes to, we tested this out. It likes to change it up on us. So no worries. Let's see, we can do the duplicate. Hopefully that'll work. Okay. Okay. Is that better?
Yes, that is
Okay.
better.
Wonderful. Let me move the bar here. And all right, well, let's go. So the title of this presentation again is Future Proofing Small Fruits, Climate Challenges and Mitigation. And when I started here at Washington State University over 10 years ago, I really didn't imagine I would be focusing so much of my time and energy on heat stress mitigation. But that really has become a strong thrust of our program, particularly for blueberry and raspberry, which is on the left and the right. And these two pictures were taken during the 2021 heat dome, which many of you probably remember that event. I'll talk about it a little bit more. But since then, we've had several other severe heat events that we've have experienced, really kind of necessitating for us to think about adaptation so that we can ensure the production of our many different crops, small fruits included. So, I want to spend a little bit of time just talking about climate change. I think many of us are probably familiar with atmospheric temperatures being on the rise, and previously we would be calling climate change global warming, but the new term for us to describe it now is climate change. And that's because although we are seeing this overall trend in increasing atmospheric temperatures, which we can see on the left, it's not leading to simply warmer weather conditions. What we're seeing is a lot of intensity or higher frequency of intense extreme weather events. And so that basically is, we're having hotter hots and colder colds, or an increasing amount of them at unusual times as well. This also is manifesting into more variable precipitation patterns. So if you think back here in Washington, we have a snow drought because we had very low snowfall in our mountains. I'm familiar with drought throughout many parts of Washington state, including on the peninsula. And with drought, we also have increased risk of wildfire, smoke, and consequences that have impact not just to our agroecosystems and greater ecology, but human health as well. So you might also be aware of some of the floods that we've had within the western part of Washington state. So this instability in our climate has also been driving more frequent once in a century events. And the flooding that we had back in November, that was a 100-year flood event. However, less than 5 years ago in Northwestern Washington, we had another 100-year flood event. So essentially, we had two 100-year flood events within this time span of 5 years, really pointing at this fact that with climate change, we're having more one-century events with that unstable climate. And this has direct impacts on agriculture. We're going to be focusing on heat stress within this presentation, and that heat stress can lead to reduced crop yield, reduced crop quality, and those are going to be key points within my presentation. But I do want to point out that when we're having these changes in our weather, because of climate change, we also can see altered pest and disease pressure as well. And this paints overall this need for adaptation strategies. So we talk about climate change sometimes on a very global scale, but I do want to zoom in a little bit and talk about Washington State and heat concerns specific to blueberry. And this is what this figure is trying to demonstrate. What we're looking at here on the left is mid-century frequency and magnitude of annual heat stress exposure over a 10-year time span for the period post-bloom or following petal fall all the way through harvest. And just take a look at it here, just because it takes a while, I think, to digest, or certainly it took me a while to digest. On the y-axis, we have frequency of heat stress in years, and it goes from 0 to 10. And this is mid-century under the most climate model where there's the most consensus and agreement on. And then we have the months of May, June, July, August, September, all the way through October. And that's a large period of time for post-bloom all the way through end of harvest, but we're thinking about all of Washington state, so the western portion as well as the portion east of the Cascade Mountain ranges. And then we have counties where we have significant blueberry production. So if you'll see, if we look at the months of, say, July and August for counties like Adams, Grant, Yakima, Benton, these are counties east of the Cascade Range. With the color coding, we can see the number of heat stress days that are predicted in the post-bloom period. And we know the heat stress days, so we're connecting heat stress temperatures or temperatures above our threshold within a 10-year period for these different counties. And they do increase substantially for these counties in eastern Washington. But when we look at western Washington, we're seeing an increased number of them as well. Just painting the picture that You know, based on the models, we're going to see an increased frequency and intensity of heat stress events for blueberry as well as other crops. Now, I want to touch a little bit on the physiology of heat stress. And I like to just think of it simply as heat stress causing indirect and direct damage to our crops. So on the left, we're looking at, a diagram just showing basic physiological principles that are occurring in our small fruit crops like this raspberry plant. So you'll see on the left, we have photosynthesis. This is a process where the plant takes energy from sunlight. It takes in carbon dioxide through those pores on the leaves called stomata. And then through the process of photosynthesis, it creates sugars for that plant, which is really important for its growth and metabolism, as well as exporting those sugars to fruits that we like to enjoy, as well as for that plant to support its growth. When those stomata or those pores are open, we have gas exchange. So again, carbon dioxide coming in, that will become, eventually sugars that fuel that plant's growth. But it also allows oxygen to escape, which is a byproduct of photosynthesis, and water vapor. So if you look at the roots, the plants are taking up water through the root system, and it moves up the body of the plant and eventually exits out of those stomata, leading to transpirational cooling, as well as movement of water throughout that plant body and nutrients that are dissolved in the soil water solution that then move into the plant. A lot of those nutrients being really essential. So this water vapor leaving the leaves through the stomata helps the plant naturally cool themselves. It's a lot like if you sweat, you get that evaporation from the moisture, it leads to a cooling effect. Now, when plants get really hot, or if there's drought conditions, or a combination of both heat and drought, those stomata will close and the plant loses its ability for transpirational cooling. And that's when we start to see scorching or that browning of the leaves that we saw on the initial slide. We'll look at that type of damage as well on some other slides. Now, that's indirect damage. That's really affecting the physiology of the plant, and that can cascade then into direct damage, such as the browning or the scorching of the leaves, because if the plant can't cool themselves, the leaves essentially cook, and that brown coloration is damage. It's basically tissue death. We can see direct damage on the fruits as well. So the image on the right is from my colleague, Dr. David Brylet at USDA ARS. And this is a study his former student, Joy Yang, did. And she, in this picture, just documented the type of damage that we see in blueberry. It can be necrosis, which we're seeing in here, A and B. It's the sunken tissue or brown tissue, and necrosis just means death of that tissue. We also can see spotting, which we're seeing here in panel C. We also can have shriveling at green and blue stages of fruit development, which D and E shows. And then we also see just poor coloration of berries, blueberries, raspberries, grapes, etc. So that's the direct damages that we can see from heat. So, we have been seeing the negative effects of heat stress within the Pacific Northwest, Washington State included, but I like to talk about the Pacific Northwest because we are really integrated with our collaborators from other institutions such as Oregon State University and USD-ARS. So our response to the initial heat dome when we saw these high temperature events and subsequent crop damage was to form a project team, and we call ourselves the Beat the Heat Project Team. We're an multi-institutional team of researchers, extension specialists, students, and postdocs, many of the individuals that you see here. And our expertise includes fields such as horticulture, physiology, engineering, breeding, economics, and extension, all really integral when we're trying to study these complex problems in perennial crops. The first project that we worked on within this team was the Beat the Heat Raspberry project. So in the next few minutes, what I'm going to do is highlight what we've learned on this project, where we're exploring promising strategies to reduce heat damage in raspberries. So you'll see the individuals listed on this slide that are part of this project team. I want to specifically call out Gilmore Menashe Makonya. He's the postdoctoral researcher that has executed all of these projects in my program. So let's talk a little bit about raspberry in the Pacific Northwest. The Pacific Northwest, including the western coastal part of British Columbia, Washington, and Oregon, has a Mediterranean climate where we're actually quite cool and moderate relative to other areas within continental North America. And that Mediterranean climate, where our summers are actually cool relative to continental climates or other type of climates, has been ideal for growing high quality fruits for both fresh and processed markets. In northwestern Washington and Whatcom County, we are the national leaders of processed raspberry production. This is really important crop for the fabric of our rural economy in northwestern Washington, specifically Whatcom County, where about 99% of the processed raspberry is. And you'll see images of how this crop is machine harvested and it's subsequently, frozen or processed. But the key thing is that for this crop, it has to be high quality for local and export markets. In 2021, we had our first heat dome event, which was really problematic for a lot of producers across the Pacific Northwest, where we had this high pressure system just sit over our region and lead to temperatures that were well above the hundreds in many locations. And that was really problematic. And that was really what I kind of envisioned as this catalyst for us within the Pacific Northwest region to, understand that we really need to study this and develop adaptation strategies. Our temperatures reached up to 117 degrees Fahrenheit in different parts of the raspberry production area in northwestern Washington, and the industry had on average anywhere from 30 to 40% crop loss due to heat damage. Some of that was indirect damage where the plants weren't simply as productive because photosynthesis and water movement was hindered. Some of it was direct damage, like the fruit here on the lower right, where we had, that white coloration, that's UV damage or scorching, and then the browning is necrosis due to heat as well. Some farmers, though, reported total crop loss, and it was about a $20 million loss in revenue for raspberry within the Pacific Northwest. So that brings us to the objectives of this project is to try to again develop adaptation strategies for the raspberry industry. And our first objective was to evaluate the impacts and cost benefits of heat mitigation technologies across several cultivars or varieties of raspberry. And we also wanted to look at different selections of red raspberry that showed some tolerance to heat stress so that we can learn more about them and subsequently they could be utilized by breeding programs that are interested in building heat tolerance within their germplasm. But for the first objective, we were particularly interested in looking at the impacts of heat protectants or biostimulants and overhead cooling and shade cloth. And I'll talk about those in a little bit more detail later on. The second objective was to examine the physiological and genetic mechanisms that contribute to heat tolerance across cultivars and advanced selections of raspberry. And then lastly, we wanted to ensure that we're disseminating project information to growers and the supporting agricultural research, crop consulting, and extension community. So it was a really big project. We're still learning from this project, even though this specific project has ended, and we're still working on our papers. One just came to me today for me to review for objective two. But I'm going to be focusing on the first objective because this is the most practical, I think, for growers be interested in learning about heat stress mitigation. So let's talk first about biostimulants or heat protectants. The reason we're looking at them is because they're a relatively low cost and potentially more immediate solution to mitigate heat stress. These are products that are being sold commercially through, cooperatives as well as agrochemical suppliers without a lot of information about them. These typically are products that you can spray on the plant or inject through a drip system and when you're fertigating and include anything from osmolytes like proline and glycine betaine, which help with plants maintaining membrane stability. It can be phytohormones, signaling molecules, trace elements, clear and edible particle films, a lot of these types of materials that claim to just improve the tolerance of the plant to heat stress, whether it be by improving the membrane stability, helping make sure that proteins and enzymes are functioning as optimally as possible because heat oftentimes leads to problems when it comes to those types of biomolecules, as well as helping eliminate free radicals through increased production of antioxidants. But research is limited, and a big question that growers have for us is whether or not to use these products. So our first trial with biostimulants was a greenhouse trial where in 2023 we took three different cultivars of varieties of raspberry, Meeker WC2188, which is now Cascade Legacy, and then an advanced genotype from the USDA ARS breeding program, Oris 47015-2. And we potted them up in a greenhouse. We irrigated them to make sure that they were at field capacity and that any subsequent differences that we saw due to our treatments was not due to drought stress, but it was due to the heat stress that we were going to impose. So here's a little bit about the experimental design and what we used for this experiment. We had plants that were, untreated. We called that our control. And then we had three different biostimulant treatments that were commercially available. We picked these three products because they represent a range of different modes of action. And growers and crop consultants said, I think there's something to these products. It wasn't very scientific, but they said, we use these products, we've recommended these products, and we think that there is benefit to them. So those were the products that we included at the high label rate. And so Fruit Armor, it's a glycine betaine. This is a type of free amino acid that is meant to improve tolerance to heat stress. Opticil similarly, and then Kelp Express as well. It's a factorial experiment. So we had our three raspberry genotypes, three biostimulant treatments, and then our control was just treated with water. And we applied these products weekly. And then we, being Menashe, my postdoc, collected data on some of the physiological variables that would indicate heat stress or mitigation of heat stress and biomass, which is another way of saying we looked at the plant growth. So let's first look at the glass house conditions. So the glass house did exceed 100 degrees Fahrenheit on numerous days. This figure, we're looking at temperatures in degrees Celsius on the y-axis, relative humidity, and then time on the x-axis. And then the red dotted line is the temperature in the glass house. And I'm showing this just so that you can see within the time frame that we did this experiment, which is shown in the dotted black box. We did get temperatures that would elicit heat stress, and those are temperatures above 100 degrees, shown by the dotted line. So we got heat stress in there. Now the next question is, did these products have any promise? Did they help improve the tolerance of the plants? Well, let's look first at biomass, because if a plant grows well under these conditions, or if there's any differences in growth, we can connect that potentially to our treatments. So we're looking at here is the total biomass of these plants at the end of the experiment, and that's both the shoots and the roots. The figure on the left is again total biomass on the y-axis, and then our different heat protected treatments, control being the untreated one in C, FA being our Fruit Armor, OP being Opticil, and KX being Kelp Express. And then we have our different genotypes or varieties and the different colors of bars. So first of all, what we saw is that Meeker and WC2188, shown in black and the dotted gray, overall had greater growth. It was more tolerant to the heat compared to the WC2188. And that's not uncommon. We do see differences based on genetics. And that, again, that's great information for us to know that these two genotypes are more tolerant to heat stress, and that can go back into the breeding pipeline as we try to build more resilient varieties with our breeders. When it comes to the biostimulant treatments, what we saw is that Fruit Armor across all the genotypes did lead to increased growth relative to our untreated control, whereas our Opticil and Kelp Express were intermediate. So that was interesting and promising. Another thing that we measured, and it's a really quick and simple thing to measure, is chlorophyll fluorescence, FV over FM. And all this really tells us is how efficient the plant is at converting light energy into photochemical energy for photosynthesis. The higher the value, the better. It indicates the plant is better able to engage in those light reactions for photosynthesis. So what did we see? Well, Meeker had greater chlorophyll fluorescence under the control, which is shown here, and across all of the other treatments. Again, showing that Meeker is a pretty tolerant cultivar when it comes to heat stress under the conditions of the study. Well, what about the biostimulant products? Fruit armor, as you can see here with my cursor, it maintained FV over FM or chlorophyll fluorescence across all genotypes. And we can see that because they all share the letter grouping A. And Help Express led to greater FV over FM in Meeker and WC21A only, but not Aura. So again, we're seeing differences by genotype. The real interesting thing for me, though, is when we look at the fruit armor, we're seeing our chlorophyll fluorescence above zero point 0.8, and that's kind of the cutoff for physiology studies. If we're below that cutoff, it indicates that that is a stressed plant. If we're above that cutoff, that means that that plant is functioning well when it comes to its photosynthetic light reaction. So this is some, again, interesting physiological data that shows that this product is improving this parameter of photosynthesis, and it's doing it better than Opticil, and it's an intermediate for Kelp Express. So that's a little snippet of our biostimulant work in raspberry. Some overall conclusions and next step for this part of the study is that, again, we saw greater tolerance or thermal tolerance, as we call it, in Meeker compared to the other different genotypes. We saw too that this commercial product, glycine betaine, found in fruit armor, led to improved thermotolerance. And we saw that through our increase in overall biomass or growth compared to the control. And I showed you just a snapshot of the photosynthetic data with the chlorophyll fluorescence. But we saw other parameters consistently also show that the fruit armor improved tolerance and photosynthesis and physiology relative to the untreated control. And it also had greater anthocyanins in Meeker and 2188. And those anthocyanins are really helpful in terms of like quenching free radicals that are a product of stress like heat. The kelp was comparable to glycine betaine, not as consistent, but still better than the control for sure and the Opticil. So that was a very controlled study that we did, but it was a good screening, I would say, for us to do a low cost study to see if there's any potential or justification to scale up this research. Now, where we're at is field trials. I just had a meeting with Menashe, my postdoc on this project today. He is now looking at application rates and timing in raspberry and different methods of application in Eastern Washington, where we consistently can get heat. So I'm not at the point of recommending these products, but I think we have a lot of promise with these biostimulant products. And I think you'll see in some of the other information that I present that this is not a one-off. We're seeing some good application here in some of our early work. But now I want to shift gears and talk about a different, more tried and true method for heat stress mitigation in fruit crops. And that is overhead cooling with micro sprinklers, which helps with evaporative cooling, and then shade cloth. So this is a project that, again, we did in Eastern Washington where we wanted to look at these two different technologies in raspberries because raspberries has traditionally been grown in cool climatic areas that is not prone to stress like heat. But we're starting to see that more frequently, and we know more frequent and intense heat is projected under these climate change scenarios. So we wanted to study more about these techniques and technologies in raspberry. So a little about the experimental design for this study. We had a raspberry planting that we planted in spring 2023. It's called a split plot design where it allows us to look at two different variables essentially. And our first variable was our cooling treatment, and we put that in our main plots. We looked at overhead cooling with micro sprinklers. We looked at shade cloth, and then we had our untreated control. A little bit more about the micro sprinklers for evaporative cooling. Those are actuated when temperatures reach 90 degrees Fahrenheit or greater. That's when it elicits a heat stress event. In the first year we did the study, the overhead sprinklers operated continuously until 7 p.m. And then that was 2024, and then 2025, and now 2025. We have sensors that turn the micro sprinklers on and off at these set temperatures. So turning on at 90 degrees Fahrenheit and then at 86 degrees Fahrenheit, it turns off. So we're reducing the amount of water that these systems are using. And then the shade cloth was 40% PAR, photosynthetically active radiation. Now, split within that, we wanted to look at how these different varieties or genotypes respond to heat stress. So we looked at Meeker and Wakefield, two commercially relevant cultivars of raspberry, and then two advanced selections, again, WSU 2188 from Washington State University's breeding program, and Oris 4715 from the USDA ARS program in Corvallis. And again, we did this study in Prosser, Washington, and that was very specific. We knew if we were growing raspberries there that we could get heat stress so we can study these treatments and their subsequent effects. So let's first look at fruit surface temperature and air temperature to let us know how well these treatments did at keeping the fruit cool. So here what we're looking at is a figure where we're looking at berry temperature. So what we did in this study, or what Menashe did, is he inserted these thermocouples on the surface of the fruits throughout the ripening period and measured their temperature. And what we see here on the figure here, we have our temperature in degrees Celsius and time. This is for a period of time in 2024 where we were having a heat wave through the region. And then we have our different treatments and air temperature. So again, this is in Celsius, but what I did here is the red line is 100 degrees Fahrenheit. I think it's a good reference point for us all. And you can see air temperature during this heat wave event did reach about 100 degrees Fahrenheit or slightly above, as you can see on the 10th of July, during this time period. So that led to overhead cooling with the micro sprinklers being actuated. And you can see that for the most part, shown in the blue dots, that overhead cooling was effective except for these 2 days right here. And what happened, and what continues to happen in this region, is that we had a mandatory water shutdown because of lack of water in the district that we were on. So even though we were at conditions to cool because the temperatures were high enough, we just physically couldn't because of upstream restrictions with water availability and water usage. And that's an important story, I think, as we think about mitigation. How available is water and how much water will these mitigation practices consume? The the shade, on the other hand, which is in green, did maintain temperatures close to air temperature, which is what we want. And then the control without any mitigation was well over 10 degrees greater than the air temperature. And that's where we start to see heat damage. So how did yield and fruit quality be affected by these treatments? Well, what we saw is that shade and overhead cooling overall increased yield relative to our untreated control. And we saw that both in 2024 and 2025. And that's what these figures here show. We got yield in kilograms per hectare for our control, overhead cooling, and shade cloth. The different colors represent our different raspberry genotypes. The big letters show us the difference, the statistical difference between our different heat mitigation treatments. So in 2024, overhead cooling and shade cloth were statistically the same and led to greater yield compared to the untreated control. In 2025, interestingly enough, the shade cloth was slightly better in terms of yield relative to overhead cooling, and it was significantly greater in terms of yield output relative to the untreated control. What I think happened with the overhead cooling is that we had even more days where we just physically couldn't overhead cool because of water restrictions. So the benefits of overhead cooling were reduced because we couldn't cool. Now, with all that water, though, with overhead cooling, we were really interested to see what would happen to the fruit quality. And what we saw, fruit quality was overall maintained with the exception of incidence of mold. So we took these fruits after harvest and we put them in cold storage and we would check on them every few days to see what the incidence of mold was. And you'll see for Meeker and Orris, which is shown here on the bottom picture here, or Orris 47015, overhead cooling did lead to significantly greater amounts of mold. So it just, and that was just the one genotype as well as with Meeker. So certain varieties have firmer fruit, better integrity, and are less susceptible to molding with overhead cooling. But the fact that we did see more overhead cooling with more rot with overhead cooling indicates that susceptible genotypes should be avoided and that free water can increase post-harvest storage pathogen pressure. All right. So on this project, I mentioned that there were economists involved as well. And so what our economist, Suzette Galinado, did is she did a cost benefit analysis where she looked at shade and overhead cooling compared to our control without mitigation. And so what we're looking at here is dollars per acre for our shade cloth and overhead cooling. So you'll see with shade cloth, our added income is much higher than overhead cooling because the benefits, particularly in 2025, measured in increased yield were much higher with shade cloth relative to overhead cooling. There were costs though, and that's kind of shown as we look at the line here below zero. So when it comes to added cost, there was labor costs when it came to our shade net, particularly deploying the shade net as well as fixing it if there were any wind events. But there was also quite a bit of labor costs with overhead cooling, and that was labor associated with weeding. So we had a lot of weeds in our overhead cooling plots because all that moisture that was cooling the plants also cooled the weeds. And we have data to show that the weeds were increased with overhead cooling. Other added variable costs too. Again, here we're looking at kind of like infrastructural costs, and fixed costs associated with the overhead cooling. But the takeaway was even though shade cloth is overall more expensive when we think about the infrastructure and fixed costs, it led to greater yields, and both technologies were more led to increased profitability relative to the control. There's a few other considerations too to think about when considering either of these two technologies for raspberry production. So when it comes to shade cloth, some of the benefits include that can dual function as hail netting, and that's become really important in parts of Europe where they're dealing with heat and unusual hail events during their fruit ripening and fruit development period. So that can be a boon, and even tree fruit growers have benefited from shade cloth providing some hail protection as well. It was the most effective at mitigating physiological indicators of heat and water stress. I didn't provide that data, but that was something that we observed in the study. And it also reduced the amount of ultraviolet radiation through its shading effect, leading to reduced sunburn. Cons, it's incredibly expensive to install. You can see anywhere from $9,880 to $12,000 per acre, and it was expensive to maintain. Several times I was out there and we had to fix it with Menashe because of wind damage, for example. And that led to, again, quite a bit of repair and maintenance costs. If we're harvesting using over-the-row machine harvesters, that type of shade structure can be incompatible with that type of farm equipment or require different types of shade netting systems that could lead to added cost as well. And then we had this concern that it might delay and impact fruit quality, but we really didn't see that when we looked at our fruit quality variables. I didn't present on that because it really wasn't that interesting. When we looked at fruit quality, we saw there were differences by the variety as well as the harvest timing, not due to our treatments. Now let's look at overhead cooling. So some of the pros is that it is less expensive relative to shade cloth, and it provides cooling and some protection due to heat. Some growers also are interested in chemigation, whereas they have these risers with overhead water being applied. They can also inject chemicals like pesticides or foliar fertilizers. So it gives them that flexibility if they're interested in that technology. Some of the cons, access to quality irrigation water may be limiting in some areas in the present as well as in the future. We saw increased fruit and storage rot in certain varieties, and that can decrease shelf life. And that's really important if you're a fresh market grower and you have to keep that fruit for a longer period of time relative to a processed grower. And then we did also see increased weed pressure with our overhead cooling. So it cooled the plants, as I said, and the weeds also benefited from the cooling. So some of our next steps for this project and our conclusions, just summarizing the raspberry work, is that we do still recognize that breeding for heat tolerance is an important and long-term goal. And so we identified, some heat physiological indicators and potential markers to help with our breeding program. When it comes to shade cloth, we saw that it promotes crop growth and offers some economic benefits, but it's largely incompatible as designed with machine harvest. So for process growers that are machine harvesting with over-the-row harvesters, it's really not a technology that's well suited for them in its current iteration. Overhead cooling with the micro sprinklers was cost effective and reduced heat related damage, but um, you know, in my opinion, moving forward, it has to be paired with smart actuation systems or smart technologies that can sense environmental conditions and turn on and off so that we're not providing excess water, particularly when we think about water shortages and water limitation. And then biostimulants, I still, I think what we saw in my first experiment was that biostimulants are promising alternatives and they're really appealing, and that's why we're continuing to do work with them. I think the real beauty of these biostimulants is that they don't require any additional infrastructure because they can be injected through a fertigation system or applied with a sprayer, and a lot of growers already have those devices on their farms. And then next steps, as I mentioned, is to do further testing of biostimulants, and we're also testing smart overhead cooling actuation systems, which uses temperature sensors as well as leaf wetness sensors, and uses that data to determine when to turn on and when to turn off our overhead cooling. So again, we're not over applying. So that's our raspberry work. In the final few minutes, I want to talk about another project that's within our Beat the Heat arsenal, and that is looking at ways to mitigate heat stress during pollination. A lot of the research focuses on fruit because fruit are really vulnerable to heat stress, and fruit ripening and harvest coincides when we tend to have the hottest periods within our year. But we also are seeing high temperature events during the bloom period for blueberry in particular. And so we have a project with the Northwest Center for Small Fruits Research where we're looking at ways to protect blueberry flowers from heat stress. Our objective one is to look at overhead cooling. So again, those micro sprinklers and see how it affects flower temperature, honeybee activity, pollination outcomes, and fruit quality. The second objective, we're looking at the effects of different ground covers like black weed mat, sawdust, and bare ground. The third objective, we're looking more at biostimulants and seeing if they have any impact on pollen viability and pollination outcomes when exposed to high temperature events. And then last but not least, of course, we want to do those cost benefit analysis and share our information with the industry. So what I'm going to do though for this project is I'm going to really focus on objective one and objective three. So that's again, overhead cooling in the field using those micro sprinklers and our biostimulant work. And for this project, I want to acknowledge Micah Evault, who's a master's student in my program. He's been leading this work in collaboration with Dr. David Bryla at USDA-ARS and Dr. Rufus Isaacs at Michigan State University. But I really want to take a minute and talk about how heat and weather is important when it comes to pollination. And I like to think of this in the context of the pollination triangle. So if you're familiar with plant pathology, there's a disease triangle. Well, we also have a pollination triangle. So if you look at the top, we have the sun representing environmental conditions, and we know the environmental conditions can affect the phenology or the development of our crop, such as blueberry flower timing onset, duration, and how compressed or extended that bloom period is. It also can affect the ripening physiology and phenology as well. Weather can also affect insect pollinators. High temperature events can lead to reduced bee activity for certain species. Cold weather can also lead to reduced bee activity. And of course, we need our bees to interact and pollinate our flowers. And so that's what that bottom line is showing, is that the weather conditions can affect bee and flower behavior and phenology. And so it's important for us to understand how these interact so we can mitigate from the negative effects of environmental conditions or extreme conditions such as heat in our case. And we know that pollen is damaged by heat. Jenna Walters, or I should say Dr. Jenna Walters now, is a former PhD student at Michigan State University in Dr. Isaac's lab, and she did really some of the seminal work to demonstrate that pollen of high bush blueberry, which is a type of blueberry we grow, is damaged from high heat. And she showed that pollen performance declined and is non-recoverable as temperatures increase from 86 to 104 degrees Fahrenheit, and she identified this threshold of 87 degrees Fahrenheit. And above that, that's when we start to see reduced pollen viability. And that's what the figure on the right is showing. We're looking at mean pollen tube length. So that's after the pollen tetrad germinates and we get a pollen tube. That's really vital for releasing sperm cells and subsequently leading to fertilization and berry development. And so you'll see as we go from cooler to warmer temperatures, there's a sharp reduction in pollen tube length as well as germination, which I'm not showing here. And that really just shows that extreme heat events can reduce pollen viability and functioning. And what I'm not presenting on, so you just have to take my word on it or read some of these papers, it also can have impacts on the nutritional content of pollen, and that can have downstream impacts on bee survival because of reduced pollen nutritional composition. And I'm happy to talk more about too if that's of interest. Um, so here's a few more images too. These are images that Micah generated of heat damaged pollen from his experiments with Aurora. So on the left, this is a healthy blueberry pollen tetrad. This blueberry pollen tetrad, we call it a tetrad because there's 4 pollen grains. It's germinated. We've got a nice, beautiful pollen tube, and this was at temperature conditions that were about 70 degrees Fahrenheit. In our heat stress pollen exposed to 95 degrees Fahrenheit, you'll see that there's no pollen germination, or though that does germinate, it looks like it's deformed. And that's probably because the membranes that make up the cells were disrupted, or the protein was also disrupted. So we just don't have normal, healthy growth from those pollen tetrads. So for Micah's work, he's looking at the effects of overhead cooling on flower temperature, honeybee visitation, pollination outcomes, and fruit quality. And as I mentioned previously, he's looking at the effects of different ground covers, such as the black weed mat that you see here in the picture. And then last but not least, as I mentioned, he's looking at the impacts of biostimulants on pollen viability and pollen outcomes after exposure to heat. So let's talk about his field experiment where he's looking at the first objective. What we did in 2025 is we established our experiment in a 3-year-old blueberry research field site in Corvallis, Oregon. We did this study in Corvallis because we had a higher likelihood of experiencing high temperature events that would allow us to study these treatments. This was what we call a split plot design with 4 replicates. So our main plot, which is shown in the red and the blue, is cooling with overhead micro sprinklers or no cooling. And then split within that, we had different varieties or cultivars of blueberry: Duke, Draper, Cargo, and Aurora. Aurora being a very heat sensitive type of blueberry. For our overhead cooling, we had our overhead cooling actuated at 80 degrees Fahrenheit, lower than what we would expect to cause damage. But again, we don't necessarily know when we do an experiment if we're going to have heat damage. So we were conservative in our actuation temperatures so that we could study these effects on some of our variables. And these micro sprinklers, after they were actuated, they operated for 20 minutes per hour. And they turned off when temperatures got below 80 degrees Fahrenheit. And you'll see that these, micro sprinklers deliver 24 liters per hour and 11.5 feet actually, I think it's 24 gallons. Yeah, 24 liters per hour, 11.5 feet per spray radius. Sorry, we represent in mixed units because academic events, we have to use Celsius and SI units, and then we use imperial units when we're talking with our stakeholders. But moving on, what we also did is we measured flower temperature using thermocouples, just the same that Menashe used to measure raspberry fruit surface temperature, and this was inserted inside the flower. We also measured honeybee activity immediately following overhead cooling, and then some of our horticultural variables such as fruit set, yield, fruit quality, and we also looked at incidence of storage rot. So let's look first at how overhead cooling did when it came to affecting flower temperature. That's what the figure on the left shows. We're looking at temperature on the y-axis in degrees Fahrenheit, and we have picked one representative day during our pollination period for 2025. This is May 5th. And the black line represents our ambient air temperature. Our orange line represents our flowers that were not cooled, are our control flowers, and the blue line represents the flowers that were treated with overhead cooling. And you'll see that when we have our overhead cooling actuated, the temperature is colder or cooler during the actuation period, but it does rise up, and at the actuation period, it's cooled down again. So you'll see this cycle, and that coincides when it's turning on and off. And we estimated that the cooled flowers were about 7 degrees Fahrenheit cooler than the control or non-treated flowers. And we could also see that with our thermal imaging as well. And so here we're able to see in the darker color where our micro sprinkler, where the plants on the canopy were cooler, shown in this darker color, relative to the plants right adjacent to it that were not getting that overhead cooling. What happened to honeybee visitation? This was perhaps the variable I was most interested in. We found that overall, no effect. So here we're looking at honeybee visits per observation, averaged across the whole period in 2025 for Aurora, Cargo, and Duke. Again, we saw no effect statistically. Draper, however, overhead cooling did lead to a reduction in bee activity. And we don't have a good explanation as to why. So this year, Micah is looking more at bee behavior to try to see if there's any behavioral attributes that the bees are engaging with when it comes to Draper after overhead cooling, or leaf characteristics that might lead to bees augmenting their visitation activities. For example, maybe on Aurora, Cargo, and Duke, there might be leaves for bees to kind of hunker down and hide under during the overhead cooling period, and they're not present in Draper. So if this is something you're interested in, stay tuned because we're really curious about Draper and why honeybees were less likely to visit Draper flowers after overhead cooling when we didn't see that effect in our other cultivars. So Micah collected a lot of data last year, and here's a quick summary of what he learned when it comes to our significant effects. We saw that flower temperature and honeybee abundance, as I mentioned, did lead to differences where overhead cooling lowered flower temperature as we wanted, and it lowered honeybee visitation or abundance in Draper only. We also saw for our sugar to acid ratio, overhead cooling increased that parameter, indicating that, that was a more flavorful fruit, theoretically, based on these metrics. But all of their variables, such as yield, firmness, fruit diameter, all the way through storage route, we didn't see any significant effects. So lastly, I want to talk about the biostimulant work that we did. So this slide is just a reminder that research is incredibly limited on biostimulants. Growers are continuously being told about these products, encouraged to try these products, but we don't have a lot of information. We're just at the cusp of generating that information, and the research is even more limited when it comes to their effects, potential effects on pollen. So Micah is doing work where he's looking at the effects of biostimulants on pollen germination and pollen performance through a greenhouse and growth chamber study. He's looking at two factors in a factorial design. First factor being our biostimulant, and we're using the similar products to the previous experiment with raspberry. Glycine betaine, which is fruit armor, and kelp express, and an untreated control. And then we have our heat stress that we impose in the growth chamber, all the way from 75 to 85, 95, and our untreated control. And in this case, again, we used Aurora, which is our heat-sensitive blueberry cultivar. So some of the variables that Micah measured after our heat stress, which was about a 3-day heat stress event simulating the heat dome that we had in 2021, he collected pollen and he measured pollen germination and pollen tube length. Those are indicators of healthy pollen. And then he also looked at pollination outcomes. So he collected pollen from untreated plants and then applied that pollen on the heat treated plants, flowers, and then he looked at pollination outcomes such as fruit set, berry weight, and seed set. So what we saw, well, when it comes to pollen tube length, fruit set, berry weight, and seed set, we saw no significant effects. So we were kind of bummed out. We always like to see significant effects as research scientists. But then when we looked at pollen germination rate, we did see our high temperature treatment of 95 degrees Fahrenheit, kelp express shown in green and fruit armor shown in orange, did increase significantly pollen germination relative to our untreated control. So I think that's really promising. We have to repeat the study to see if that's consistent. But this is one of the first studies that we're seeing a beneficial effect or where someone's actually measured the potential effects of these biostimulant products on pollen performance. So just some conclusions from this work, because I would still say it's preliminary. We have 1 of 2 years that we've been doing this research. We were able to demonstrate what we have known from Dr. Jenna Walter's work. Extreme heat reduces pollen viability and impairs reproductive processes that are critical for the formation and development of high quality fruits, including blueberry. We saw overhead cooling lowers flower temperature without any negative effects on yield or storage, but it might reduce honeybee visitation in some cultivars, and we saw that in Draper. kelp and glycine betaine or kelp express and fruit armor, those biostimulants improve pollen germination under high temperature conditions of 95 degrees Fahrenheit. And we're right in the midst of doing our follow-up experiments for 2026. Micah just applied his biostimulants 2 days ago, and we're going to be getting him in the growth chamber on Friday. So just as a wrap up, because I talked about two different projects that are within our Beat the Heat climate change adaptation arsenal, just want to leave you with some final thoughts before we go to questions and discussion. So, when it comes to my program and many other programs interested in stress tolerance, we're already operating under the assumption that climate change is really going to have an impact on crop production, and that understanding and recognizing this is an important undercurrent to the type of work that we do and developing strategies so growers can adapt and mitigate the negative effects. Implementing both short and long-term adaptation strategies, I think, is going to be really critical for future-proofing small fruit production. And so some of the short-term strategies that we've been looking at include overhead cooling, which we found is effective but does require substantial water inputs. Shade cloth can reduce heat and UV to a point. If it gets really hot, like above 110 degrees Fahrenheit, we start to lose the benefit of shade cloth. But it's costly and it can interfere with mechanization. And then biostimulants, I'm still not at the point of recommending them, but I'd say it's really a promising technology, and more research should hopefully elucidate when they're beneficial, if they're beneficial, and optimal application strategies. And then lastly, I think, smart technology such as, sensors that are really effective at deploying cooling technology, so we're not, say, over applying water, as well as breeding for resilience are going to become increasingly important as we think about adaptation and start to implement these technologies. So with that, I want to thank you for your time and attention. I know it's always, I find after 5 o'clock is always a little bit late for my brain, so I appreciate your patience if I kind of fumbled through some of this. But I like to acknowledge funding from the Northwest Center for Small Fruits Research, the Washington State Department of Agriculture, and the USDA Special Crop Multi-State Block Grant Program. And I'm going to be taking my slides down in just a moment. But if you're interested in this work and the type of outputs that we're generating from this work, please do scan the QR code, visit our website. We have a lot more information and resources there, and we'll continue to put information up there as well. So with that, thank you, and I'm happy to take some questions.
Great. Thank you so much, Lisa. That was fascinating. I'd love to open it up for questions, but before we do, I just want to bring everyone's attention. We put our evaluation to this work or this event in the chat, so please fill that out before you leave tonight. But I have a question to start us off. I'm curious if there's any other methods being explored for overhead cooling that doesn't involve water, like airflow or other.
Yeah,
Yeah.
Kelly, that's a nice question. I'm not aware of any other technologies that are really focusing on evaporative cooling that don't require water. For evaporative cooling, you'll need water essentially, but there are some cultural practices that you can implement in terms of just like trying to promote good airflow in your field through placement, awareness of structures that could impede the flow of air. The one technology though that is evaporative cooling that I'm pretty excited about is fogging systems. And so in this case, you're just changing the droplet size, and you're not necessarily cooling the crop so much as you're cooling the air through evaporative cooling means. So in tree fruits, we found that this can be effective at lowering the temperature of an orchard and the temperature of the fruit, but not necessarily cutting back on water consumption because you have to operate them longer. But we'll see. In blueberry, it's a different environment, different climate, different canopy structure. So that's something that we're trying because in some cases, like grape, they have found fogging systems to help cool the fruit but reduce water consumption.
All right, looks like Tara's asking a question in the chat.
Great question. So, did overhead cooling reduce how much water was needed for irrigation? That's a great question. It can. Just with our study, we wanted to make sure that the plants were getting consistent irrigation. And so they were all really well watered. The overhead cooling did introduce extra water. So, in that particular experiment, we could have been overwatering the plants theoretically, but optimally, when we're thinking about a grower implementing these technologies, they're measuring their soil water content, they're measuring canopy and leaf temperature and air temperature, and they're adjusting their irrigation if they're deploying overhead cooling so that they're staying within a certain, you know, volumetric water content level, and they're measuring their crop and adding irrigation water based on evaporative demand.
Want to open it up for other questions? Feel free to unmute and ask away or use the chat box.
I have a quick question. Um, a lot of the growers in our area prefer organic methods. Are the biostimulants, are those typically registered organic or are they kind of both?
Laurel, for that question. They can be registered as organic products. We're using a couple of different organic products and some other trials as well. Some of the kelp-based products coming from our local, or I should say local, but from suppliers closer than where Ascophyllum nodosum, the product that we worked with, is from. And so again, stay tuned. Again, I'm pretty excited, but cautiously optimistic about these products. But those kelp-based products, as well as some of the other, even like lysine betaine, they can be USDA OMRI certified. Um, you know, I think the big thing I'd say to be aware of is well, these products, we're still not recommending them. They can be quite costly, and a lot of the suppliers recommend continuous application of them, and that can add cost if you're applying them, every 7 to 14 days. Based on the physiology of priming, and that's what these products essentially are doing, is they're eliciting a priming response. They're encouraging the plant to be more tolerant to a future stress. That priming effect, um, if you're constantly in a prime state, that can actually be really costly and expensive for a plant. It's like always being in a state of having an adrenaline rush. It's not necessarily good for your body. You're taking energy away from other things that you could be doing. So, um, you know, a lot, a lot of the questions that we have about these products is, do you really need to apply them continuously, or can you be more targeted with their application? That's kind of what the physiology of plants and priming stress indicates. So if you're interested in testing these products out, feel free to do so. They're still, I'd say, interesting, but not something we recommend per se, just based on where the stage that we're at in the research. But I would just aim to apply it the few days before a stress event, whether it's heat or cold, as opposed to continuously applying it where like what a manufacturer might recommend.
Thanks.
Does anyone else have any questions? Now's the time while we have our specialist, our small fruit specialist here tonight. But
I could ask another question if
okay.
Okay.
nobody else. Okay. So you mentioned that you may be looking at ground covers. Are you currently doing that research or uh, any kind of preliminary thoughts on that?
Yeah, absolutely. I took out some of the results that we've generated to date on ground covers, but what we did see is that our black weed mat, or that black woven fabric that many blueberry growers and organic fruit growers often use, does increase canopy temperature. So during the bloom period, we didn't see any effects on temperature because it was still relatively cool. But as we go forward in the summer and air temperatures increase, the magnitude of the differential became significant. So, when we're thinking about climate change, heat stress events, protecting fruit, bare soil, particularly if you have dark, dry, bare soil, as well as black weed mat, will increase the temperature of the canopy and increase the susceptibility or risk of heat damage. The Douglas fir sawdust mulch, on the other hand, or some of the white colored type mulches from other research trials, does a really nice job reflecting sunlight and has more of a net cooling effect as opposed to, again, dark, dry soil or the blackweed mat.
Okay, those were going to be my follow-up questions. Thank you.
Mhm.
I have another question.
Sure.
So this year we had some really early heat, I guess.
Mhm.
Um, is there any research going on of like how we can protect our plants from, the early heat, then it gets really cold again?
Mm-hmm. Yeah, so that's tricky. So, Tara, are you thinking about kind of the early heat that we had, like January, and then it gets cold in February, and kind of that late winter, early spring period? Or are you thinking about a different time point?
Yeah, mostly that.
Okay. Yeah, that's tricky. So that's also something we've been looking at. A lot of that is the genetics of the plant. So certain types of blueberry and blackberry and raspberry that are being sold here are low chill, which means, they don't need many hours at temperatures, slightly above freezing for them to complete flower bud development. And so once they meet their chilling requirement, which for a low chill or no chill type plant might be in end of December, which we saw this past year, or January, and we're not out of the risk of the frost window at that point. Um, and then if we have these warm temperature springs in January, that can drive that plant to really move forward in its development. And as it moves forward in its development, we have increased susceptibility to freeze damage. And so just I want to explain the physiology in case others aren't aware of that phenomenon. But selecting varieties that are high chill or adapted to our region or coming from breeding programs from our region, I think that's step one. You reduce the risk of that type of event happening. And then once you have that plant and you're having these types of fluctuating springtime conditions, there's not a tremendous amount we can do to slow the plant down, because light and temperature are really strong drivers for that plant exiting out of dormancy and increasing their susceptibility to freezing temperatures. But we can do some things like just frost mitigation. And frost mitigation at large scale can be like fans. At smaller scale, um, it can be things like, uh, uh, sorry, I was telling Kelly, like after 5 o'clock, I get really tired. So you're seeing a byproduct of that. So in some cases, propane burners or burning, and I've been seen in our orchard systems burning to release heat for those plants. Not that I'm encouraging that. And then biostimulants also have been explored as another tool to can prime plants so they're more resilient to freezing stress. And we've again seen some interesting like results that make us optimistic, but we're not at the point where we're recommending them. So hopefully that was a long-winded answer, but hopefully that gave you insights on the physiology of freeze damage and how these variable springtime conditions can increase the risk of frost injury. But really, genetics selecting for adapted varieties from our region is step number one, and trying to avoid things that are coming from programs outside our region because they oftentimes have lower chilling requirements, so they're ready to go at an earlier time period than oftentimes that we're ready for within our region. And then, mitigation measures, but those are largely things that are done at larger scale.
Lisa, are there any particular varieties of blueberries that you would say have those lower chill requirements that are people are starting to plant more often here that they might want to avoid?
Yeah, yeah. Peachy blue is one that I absolutely love, but it does tend to be a little bit more cold tender. The ones that I'd recommend though are kind of like the classic northern highbush blueberry varieties. For example, like Blue Crop and Jersey and Duke and Calypso. So those are coming from programs either here within the Northwest, or they're coming from programs like Michigan State University, which is also dealing with climate change and similar to what we're dealing with, but they do have frost events. So it's coming out of their program tends to be more cold, hardy, and higher chill types.
Well, I think now is a good time to call it a night and wrap up our presentation. Lisa, thank you so much. We can definitely put some information in the chat or follow up with people on where to learn more about the Small Fruits program. And if there's upcoming field days for farmers and just ongoing research, we can certainly help connect you with those resources. But thank you so much, Lisa, for your expertise tonight. And we look forward to your ongoing research.
Yeah, my pleasure. Thank you for having me. And again, I apologize if there anything was unclear. If there's any questions, people are welcome to reach out to me.
Great. Thank you. Good
All right,
night.
bye.